Okay, so I think we can start. Good afternoon, good morning. I don't know everyone. So I wish you a very, very happy New Year to 2026. I hope everything will be fine for all of us. So we'll make a little introduction about David Rouquie, that is our speaker today. So David is a toxicologist at Cropscience facility in Sophia Antipolis. He's responsible for the new form LifeHub in Sophia. He's really dedicated to science and research, especially when it is addressing social needs. So he's involved in multidisciplinary projects in his company, but he's also participating outside in his company in other projects like with HESI. He has a background in biochemistry and molecular biology, and he's at the intersection between biological and computational sciences. He's a Bayer representative and vice chairman at the EUROTOX Scientific Committee. And in the context of the Broad Institute HESI collaboration, is co-chairing the OASIS initiative. This initiative aims to build confidence in the use of cell painting, transcriptomics, and proteomics coupled with exposure modeling for chemical safety assessment using hepatotoxicity in a case study. So please, could you shut down your microphone? The presentation is recorded and then it will be available on YouTube, on the YouTube channel in EUROTOX, like all the other webinars. Please put your question in the chat, and at the end of the presentation, I will read the question to David. So the presentation is multi-omics and their application in predictive toxicology and risk assessment. Thank you very much, David, again for accepting to give this webinar. The floor is yours.
Thank you, Mark. Thank you for the kind invitation to give this presentation first. I'd like also to wish you all my very best wishes to all of you for this new year, 2026. And in this presentation, I will touch upon some of the results we have been generating internally at Bayer Cropscience, but also in some inter-industry association, namely ECTOC and HESI. So I'll skip the next slide on the CV and I go straight to the agenda about my presentation. You will see I will be referring very much about complexity and navigating the complexity. So during this talk, I like to go through this introduction on NAMs and NGRA, globally speaking. So NAMs for new alternative methods and NGRA for next generation risk assessment. Touch upon some current limitations, so do some kind of reality check. Mentioning about the case study in method development. Mark mentioned it earlier on about the OASIS consortium that I will briefly describe. And then go to some proposal of path forward about integrated testing strategy and then conclude. So when it comes to the NAMs and the next generation risk assessment, that's I like to maybe put on in perspective the two different ways of approaching these testing paradigms in chemical safety. This triangle representing somehow the different types of assays that can be performed to assess and define safe dose of use of chemicals, generally speaking. Assuming that the size of at the basis of this triangle inform about the numbers of studies or evaluations that can be performed. We start at the bottom with the in silico methods, where basically there is almost no limitation in terms of a number of evaluations that can be performed. As soon as you get the structure, you can make use of the representation in the digital space about the chemical structure. And if you have the appropriate set of tools in terms of models, then you can predict. Climbing up into the ladder of this triangle, we go into the in vitro study evaluations, then moving on into the subacute, subchronic studies in lab animals, ending on the top of this triangle in the apical studies. And historically, the way to perform risk assessment was really deriving the information mostly from the apical studies and moving into a top-down approach, characterizing the adverse effects observed in those apical studies. Then some mechanistic insights from these tox effects revealed in these apical studies would be performed. This mechanistic information would be derived, and then a human relevance assessment would be performed in order to both determine safe dose of use and inform about the safety profile of the molecule, globally speaking, for human. This is the actual testing paradigm currently still in use, and in opposition to, or in addition to this actual testing paradigm, you have the emerging one, where basically the starting point is at the bottom of this triangle. We're using in silico and in vitro methods, and you see that there is a little bit of the overlapping of this red circle onto the subacute studies. That will be a key point of my presentation today. That can be seen as one thing we need also to address even in the emerging technology, in the emerging testing paradigm. So in that next generation risk assessment, it's a bottom-up approach where those evaluation allows us to gain information in terms of mode of action, adverse outcome pathways, toxicity pathways, which combined with quantitative to in vitro to in vivo extrapolation. So basically trying to convert micromolar into milligram per kilogram per day information from the bringing us to this prediction of the safe dose of use in human. And on purpose, I put a question mark in between the qui-vive and the predictions, because there are some discussion about if we need to get still some safety factors in order to make the prediction of the safe dose of use in human, globally speaking. So in this paradigm shift, we are moving from basically the gold standard in terms of evaluation using histopathological examinations to in silico and multi-omics readouts. During this presentation, I will be very much focusing on in vitro, the use of these multi-omics readouts into in vitro cell-based assay, but also in short-term in vivo mechanistic assays. Bearing in mind that we should not forget about exposure science, in particular when we have to deal with the in vitro cell-based assays, to convert what we know about the effects at the concentration, the effects are induced in cell-based assay compared to what can happen in human situation. So this multi-omics readout, so what also called high content biological assays, have compared to, in particular, individual biochemical assays, several advantages, as they generate rich data and molecular responses for both tissues and cell cultures. They support the identification of benchmark concentration for biological activities, so the so-called PODs, point of departure, which are critical in the next generation risk assessment approach. They also enable chemical grouping based on bioactivity patterns. And they also provide insights into mechanistic understanding about how come that a molecule induce an adverse outcome, yes or no. So altogether, those assays make them valuable tools to advance the ability to conduct exposure-led science and hypothesis-driven assessment, both in early de-risking of chemicals, but also in chemical risk assessment in general. So when I'm referring to multi-omics data, I will be mostly referring to transcriptomics, metabolomics, proteomics, and I will place a little bit of the focus on the new kid on the block. I would call it like this: the cell painting assay, which has been used in the context of risk assessment quite recently. And since it's a brand new tool, I want to spend a few minutes to describe a little bit the overarching principle of this assay, the cell painting, which is an high content image-based morphological profiling assay. This assay has been the first publication is from 2016, I guess. So almost 10 years, so a very recent one. It has been developed by and invented by the Dr. Ann Carpenter at the Broad Institute in Boston. The overarching idea from the cell painting is very, very simple. Nevertheless, it's super complicated and very technically challenging to put that implement it in the lab. But the overall idea is that a molecule that gonna do an effect into a cellular system gonna influence the morphology of the cell that might not be seen by eye, but that can be seen using digital tools. And those changes are consistent across molecules sharing similar properties. So molecules having similar targets or similar mode of action tend to get morphological profiles similar in this assay. So in a nutshell, this is an high throughput assay. So it typically is run in a 384 well format. So you grow cells, could be cell line, could be primary cell culture, could be iPSCs, could be genetically engineered cells. You expose your cells with your perturbator. So it could be, by the way, not only compounds, chemicals, it can be peptides, it could be antibodies or whatsoever. So you expose your perturbator. At the end of your time of incubation, you fix the cells, and then you stain the cells with different fluorescent dyes, which are going to stain specifically different subcellular compartments of the cells, which are listed in the slide. So it can be ER, it can be nucleus, it could be the actin cytoskeleton, it could be the mitochondria. And all those different images that are derived from different stain are then used to extract, describing the distribution and the properties of the different pixels of the different images to end up with these morphological profiles. And after a fancy set of normalization and different step at the computational level, you can end up with different those morphological profiles, or can be used to cluster them and draw some conclusion out of those assays. So historically, this assay has been used for heat discovery. It's even more recently that it has been used for toxicology. The very first seminal papers on the use of cell painting for tox was done by at US EPA in the group of Joshua Harrell. And since then, there are many examples where cell painting has been used for different toxicity endpoints. So I will be referring to acute tox. There are some papers also on mitochondrial toxicity, developmental toxicity, and liver tox. So having spent some time just about specifically on the cell painting, I'd just like to move on into this a concept and a term that you will be hearing me in the next slides about spaces and in particular in vitro and biological spaces. So on that slide, you have the description about a very famous description about the way an adverse outcome in lab animals or in human can take place. So it's an image that you have probably seen many times. It's derived from the National Research Council paper from the 2007. So basically, you have a chemical on the on the left-hand side, reaching a biological system to reach some molecular target, inducing some changes at the tissue level. And then if the dose is high enough, or if the time of exposure is long enough, then it does translate into an adverse outcome in mammalian system, in lab animals or in human. So during my talk, I will be referring to chemical spaces for a set of chemicals from a given sector or a given application. I will be referring to in vitro biological spaces when it comes to in vitro evaluations in different cellular models using different technologies. It could be cell painting, it could be transcriptomic profiling, it could be proteomics. And I will be referring to in vivo biological spaces when it comes to different target tissue in different specific in a given species and a given sex, for example. And in the context of NGRA, one of the big key challenge is to make the link between those in vitro biological spaces with the corresponding in vivo biological spaces. So during, again, this presentation, when it comes to the definition of this and the representation of those spaces, I just want to illustrate that in one example. So here you see the two-dimensional space of a chemical space, where in this representation, so it's a t-SNE plot. You probably have seen also some other types of two-dimensional representation of this high-dimensional data. So it could be t-SNE, it could be UMAP, but basically the very basic principle is that each dot in that case represent a chemical structure, and the distance between the dots inform about the degree of similarity among the different chemicals. So the closer the molecule are, the more similar they are. You will have the same type of representation, similar type of representation for the bioactivity signatures. So in that case, each dot represents a bioactivity signature. That could be a transcriptomic profile, it could be a cell painting profile, that could be a metabolomic profile. And again, the same rules applies. The distance between the dots inform about the degree of similarity of the signatures. And the cool thing with that is that you can monitor between the two spaces, chemical space and biological space, how much they are related. And so in this example, I've highlighted in red a series of molecules which are structurally similar. And then you can jump into the biological space and have a look into the nature of the effects which are visible in a given biological space, and it can go back and forth from one space to the other. So in toxicological sciences, we are dealing with different kind of spaces. So we have, as I said, chemical, we have the in vitro biological spaces, we have the in vivo biological space from lab animals, and the one from in vivo biological space in human. And the thing is that there are multiple, because you will get different kind of chemical spaces depending on the sector. So the drug space will be something which can share some similarity with the agrochemical space and industrial chemicals, but they are distinct. you will get some in vitro biological spaces differing from the species of the cellular model, the cell types, if it is a primary cell culture, if it is a cell line, for example, the time of exposure, the concentration, and of course the readout. So if it is different types of omics. And when it comes to in vivo biological spaces, you will get the species, of course, if it is a lab animal or human, the genetic variation, the sex, the tissues, the time, the dose, and of course the readout. And in this context of NG array, globally speaking, the key challenge is to better understand the interplay between all those different spaces, how much they are related, how much they are overlapping between each other, how much the description of the in vivo biological space in lab animals compare to the one in vivo in human. So that's all about the translations, basically, which are taking place between these different spaces. And just to give you one example, so as Mark said, I'm working for Bayer Cropscience, so that I'm from a company which still own the two sectors from pharma and Cropscience under the same roof. So we have quite a bit of exchanges with our colleagues from pharma, and so we can share some information. So this is the chemical space. I mean, a snapshot about the chemical space from pharma and Cropscience chemistry. And from this representation, you can see that there are some overlaps between the chemistry from pharma and the one from agrochemical, but still there are some island of chemistry which are really specific on one of the other sectors. When it comes to the in vitro biological space, in that case, in different cell lines, this is a representation of 8,000 molecules that have been profiled in different cell lines, which are listed there, and that you have the patchwork of different effects induced by all those 8,000 molecules in these different cell lines. So now I'd like to move to the second part of my presentation about the reality check and the current limitation, providing some examples about what we have been doing in lab. And the first example I like to give you and present you today is about an illustration about the chemical applicability domain limitation in the context of the prediction of strong acute toxicity of small molecules. So it's a work we have been doing in the frame of a PhD thesis from Fabrice Camilleri, where basically Fabrice make use of a set of roughly 250 chemicals, roughly, with both known molecules inducing strong acute toxicity and some which are not doing strong acute toxicity.
Produits chimiques pendant sa thèse.
And what Fabrice did was to build some cases where he trained QSAR models using a training set which was representing the entire chemical space from the training set. And he did make the testing using a molecule which were part of the training set. And doing so, using the QSAR model, he managed to get some very nice balance accuracy about 0.8, indicating that the performance of the model was quite good. In opposition, Fabrice split the chemical space into two different spaces, the one dealing with the training set and the one for the testing set. So basically the model, the QSAR model did learn on a different kind of chemistry from the one from the testing set. And in that case, as expected, he did see that he did lose a lot of this predictivity, having a balanced accuracy, only about 0.6 as compared to the 0.8 obtained in the first case. The interesting aspect was that we did at the very same time an evaluation with the very same molecule at the biological level, and we did that using the cell painting and using the same approach. Instead of using the chemical structure as an input file for making the prediction. In that case, it was this morphological profile based machine learning model. What we did observe that in the case of the non-chemistry, the balance accuracy was declining a little by 0.774, but we gain the best model using the cell painting profiling in the case of novel chemistry. So it's just to illustrate you how much knowing about the prediction from the chemical structure you need to know to train the model using an appropriate space of the chemistry. And if it's not possible, then you can move into the biology and try to predict based on biological information. This was the first example I wanted to provide you. The second example I wanted to show you today was about the biological applicability in domain application. There also, it's another example from the work we have been doing in the lab. So making use of public data set, a large public data set of 8,000 molecules, which were profiled using transcriptomic tool on 4 cell lines. And I just want to illustrate what we did observe. So we are here in the biological space, and we have there the representation of the biological activity of those 8,000 molecules in 2 cell lines. The brown, in brown color, those are the result obtained from the VCAP cell line, which is a prostate cancer cell line. And in the light blue, this is PC3, which is a cell line also generated from prostate cancer. And what you can see is that you barely see an overlap between the signatures derived from this 8,000 compound in the 2 cell lines derived from the same initial tissue, which is the prostate, which was at first sight a very surprising approach. So what's revealing that depending on the cell line, even derived from the same tissue at the very beginning, the biological effects generated in those cell lines can differ dramatically. And this is another example in this exercise using 2 lung cancer cell lines, where essentially we did see very few overlap between the signatures obtained from these 2 cell lines derived from lung cancer. So this question the biological relevance and ask us the question about how much a given cellular model capture the key biological processes relevant for the question we have related to toxicity in particular. Another example about the time response limitation, where here we did compare the result obtained again using a public data set obtained with a set of 110 compounds using cell painting. So again, in vitro biological space, where what you can see, it's for each of the cell lines and each of the time points used in this with those data, we have a distinct representation of the biological space of those molecules using cell painting. So distinct according with the same cell line at different time points, and of course the same with different cell lines. And type of results using then the transcriptomics, where you do see there still have a little bit more overlap between the responses obtained across the time point, across the cell lines, but still quite distinct still. So out of it, what we did conclude is that both the technology and the measurement timing affect the biological interpretation of the result, which needs to be really taken into consideration when it comes to the use of in vitro based method for in the context of NGRA. Last example is about the importance of in vitro free fraction when it comes to in vitro cell-based assay. So in vitro cell-based assay, typically what we consider the concentration of the molecule is the one which is put in the well, so the nominal concentration that what is placed in the well of the microtiter plate if we take this example. But the thing is that it's only the free fraction of the molecule that's going to be driving the biological response to the cell to induce the biological effects. And it's just an illustration about a series of 45 chemicals for which we have evaluated the free fraction of the molecule and the fraction of the molecule which bind to the plastic of the microtiter plate, the fraction of the molecule which bind to the cellular the cell media in a cellular assay. And what you can see that you have a whole range of different situations from 0 to almost 100% of the molecule that can be found as a free fraction. And you have a huge diversity of the properties of the molecules when it comes to the level of the free fraction you can get in your cellular assay. So this to illustrate that assessing the free fraction of chemicals in cell-based assay is really vital for accurate toxicity assessment, in particular when it comes to the determination of the point of departure. Looking at the time, I'm not sure I will have time to go through the few slides. So I might skip them to spend some time more for the last bit of my presentation. So, but essentially the thing is about more and more we are recognizing that biological data, in particular with this multi-omics data in the context of cell-based assay, to restrict this both biological and technical variability is there. Globally speaking, when it comes to this in vitro multi-omics evaluation, few things is that these various omics technology describe specific layers of the universe of chemical-induced effects. And for each layer, we need to better understand several things. So the natural variability of the individual descriptors, so could be transcripts, could be set of transcript, proteins, metabolites, or morphological features. That corresponding dynamic range, and the proportion of the biological knowledge they provide with regard to the adverse outcome. So historically, we have been using transcriptomics because the technology was there, but how much transcriptomics is uncovering the biological responses from chemical-induced effects compared to the proteomics, compared to the metabolomics, compared to the cell painting is yet to be done. I like now to quickly go to the case study facing all the limitation I briefly described you before. What should we do? And one of the approach is to get insight and to better understand whose limitation, describe them and characterize them globally speaking. And this is one of the objective of this OASIS collaboration, which is an SC Broad Institute collaboration, where the idea is to try to build knowledge about the use of this multi-omics data using in vitro test method compared to what we know about the molecule in vivo. So the initial driving forces was of two kinds, from the ACHEM sector about these NGRA aspects, and for the pharmaceutical aspects, it was more a desire for an integrated clinical translatable high throughput screen for prediction of adverse outcome in patients and to prevent later stage attrition. So the vision of this multi-sector, multidisciplinary collaborative effort is to serve both the specific needs for ACHEM and pharma and build and generate useful insights in the context of NAMs. So we decided to focus on to make it a kind of simple, but still highly complex, to focus on the use case on hepatotoxicity, both as a compromise because it was of interest for both from the ACHEM sector and for the pharma sector. And the idea is to benchmark the hepatotoxicity induced by a series of compounds, well-characterized compounds, in vivo, in the rat in particular, but also in human, making use of the so-called DD compound, drug-induced liver injury compounds, and profile them against and benchmark the results from what we know in vivo with what we obtain in vitro. So we got a grant for this from the MLSC, and we have the contribution for the industrial partners. And the duration of the program is 3 years. So we are almost to the end of the first phase of the OASIS program this year, and we are thinking about the phase 2. It's a large consortium with academic and non-profits, government and regulatory bodies, and 16 industrial partners and some tech partners, so mostly startup company in the area of the Massachusetts state in the US, close to Boston. So very briefly, the overall objective is the following. We start with compound for which we know what the molecule are inducing in vivo. So I take this example in the rat in that case, and also the effects and the no effect level. And using hard cell culture, we're going to profile them, and we are profiling them using transcriptomics, determining the point of departure, estimating the equivalent dose level in vivo using the key-vivo approach, and benchmark those results, both the mechanistic information and the dose level, and using the effects with the one known in vivo. The same is done also using cell painting, and you also using human cell cultures to do so. And all this idea is to benchmark the two sets of data between in vitro and in vivo. And in terms of translation, as I said, we are benchmarking the rat situation from hard cell culture versus in vivo rat using a large set of molecules. The compound list from the OASIS compound is something 1,200 chemicals. We do the same using iPSCs, primary cell cultures, with regard to the in vivo situation in human, using the DD compound. And we also want to make some learnings between the translation between in vivo rat and in vivo human as well. So back to this representation of species in OASIS. So we are profiling mostly two chemical species, the ACHEM and the pharma chemical space, both at the chemistry and the in vivo biological space level in the liver. And we are evaluating several in vitro biological species, as I said, from rat and human, primary cell lines, and organ on a chip also. And all these things is in order to gain knowledge about the translations between those species. Take-home messages from these aspects. So we have this. So of course, the historical in vivo toxicity data is a treasure for us to gain biological knowledge and make these conceptual learnings between those data generated in vitro compared to the one taking place in vivo. Still big challenges we're trying to cover. I mentioned some of them earlier on, and through this global cooperation between academia, regulatory bodies, and industry, that's a nice way to gain this biological this information and this knowledge. And quickly, I like to end this talk with a forward-looking about taking into consideration what has been said before about the reality checks, the limitation of this in vitro test methods, but still thinking about an implementation and a realistic implementation of next generation risk assessment in the near future. And for this, I like to provide you and go through some thoughts we are developing in the context of VC Talk, but some other also in some other entire projects, European project, also at the US EPA level, about the use of short-term mechanistic studies in the context of next generation risk assessment. So if we assume this is the whole spectrum of types of evaluation represented in this on these slides that one can perform for chemical risk assessment. You will get on one side of the spectrum the pharmaceutical products, where in silico, in vitro, preclinical data in animals and human data generated in clinical trials or performed to evaluate the safety of the drugs. On the other side of the spectrum, you will have the cosmetic products, where only the in silico and in vitro test method are used for the risk assessment for systemic toxin in particular. And now the question is about where to place the cursor for industrial chemicals and agrochemical products. And so we have we know that we won't go into the direction of pharmaceutical products. In any case, we will make some clinical trials with both industrial chemicals or agrochemicals, but do we still need to go toward the cosmetic products, or can we find an intermediate position? And that's exactly what we are proposing. So to complement these in silico and in vitro methods using short-term in vivo studies, enrich multi-omics endpoints, to better characterize the risk and define the safe dose of use of those products in the future. So this approach has been published in a position paper because it was something we proposed in a session at the SOT last year. And in a nutshell, the proposal is to move toward reduction and refinement rather than complete replacement of animal testing in the future, and having the NAMs, so in silico, in vitro test methods, complemented with some smart short-term mechanistic studies that can be called SMART studies, ETAP studies, or from the US EPA or TG plus, test guideline plus studies, and to predict the context in human. So making use of the NAMs in vitro to predict human situation when we gain enough robustness in terms of the assessment, and make use of the SMART studies also to complement the risk assessment, in particular with long-term toxicity study, developmental toxicity, or more complex type of toxicity. So having in mind that understanding both capabilities and boundaries of the current method, the ethical choice to complement in silico and in vitro methods, with short-term in vivo studies coupled with multi-omics tools that maximize information while minimizing animal use. And with this conclusion and key messages quickly. So NGRA represent a necessary evolution in toxicology, but we must be realistic about the current capabilities. In vitro and in silico methods provide valuable mechanistic insight, but have defined limitations. And this understanding about applicability domains, temporal dynamics, model-specific characteristic is essential. One path forward is also the collaborative efforts like OASIS to demonstrate the values of systematic method evaluation. And current best practices involve integrated testing strategy that thoughtfully combine NAMs with short-term in vivo studies. And the goal is not perfection, but continuous improvement toward more ethical, efficient, and scientifically sound risk assessment. And with this, I just want to close and by thanking you for your attention and for all the connection and all the colleagues which were involved into this reflection and the work that I've been presenting to you today. Thanks again for your attention.
So thank you very much, David, for this very comprehensive presentation. We have many questions and we have 50 minutes to answer them. So I will start with Jonas. So regarding chemical and biological spaces, it is on the slide 13. As they are high dimensional, I would think applying a machine learning model and extracting its feature importance could be interesting to see whether they contain orthogonal data. Was this done, and what was the outcome?
Alors yes, that's exactly the thing. It's there is always the thing about the data quality and the amount of data to make use of this type of in silico tool. So having sufficient amount of data, and we have published that we can build machine learning models out of gene expression signatures. So instead of making use of like for the QSAR, structural features of the molecules, you can make use of these transcriptomic profiles. We did that also with using the cell painting profiles and train a model that learns from the bioactivity patterns, properties of the molecule, and predict some properties of the molecule out of it. And that's what I try to illustrate. Doing so, you somehow go over the chemical space limitation from the QSAR models. So you go straight in the biology. So you look at the effects, you learn from the effects induced by the molecule in biological system to make the prediction. So the short answer is yes, we did that, and it's quite promising also as a tool, preventing to get you have the appropriate set of data to do that. Yeah.
Thank you. So now I have two questions about the free fraction. I have one question from Danilo and one question from Marie-Christine. So the question from Danilo is: was the evaluation of the free fraction estimated in silico, Armitage model? And the question of Marie-Christine is the same question about the free fraction determination is challenging and depends on a lot of parameters with the percentage of the serum, etc., albumin vehicle, how estimate the free fraction. So it's about the complementary question of how did you estimate the free fraction? Did you use a model or did you use other parameters?
No, it was done experimentally. So we did measure, like you do the free fraction measurements in the blood, for example, which is an essential piece in toxicokinetic evaluation. You can do that with the cell media culture. And so like this, you really assess how much a molecule is there to induce an effect, assuming that if it is bound to a protein, mostly to a protein in a cell media culture, and even more in the case of a plastic of the cell of the microtiter plates, then the molecule won't have an effect. Won't be
just one question, just one comment. I mean, because you're doing cell culture, so you think that the free fraction is quite stable Okay. When you add it, it is stable because you have the plastic interaction, you have other type of interaction. So it's quite different from the blood. That was I mean.
Yeah, sure. But so what we have done was you expose you mix your molecule with the cell media culture, and you assess how much of the molecule from the cell without the cells, how much of
Without
your
the cells and without the plastic.
in the plastic itself,
Okay.
in the cell, from the microtubule. Yeah, you can do that. Yeah.
Okay. Then another question from Danilo. What was the choice of selecting proteomics over metabolomics, which could provide a closer link to the phenotype?
I would say that is yet to be defined and yet to be demonstrated. I would say that historically, again, the data that has been generated in the omics field in tox was historically the transcriptomics. For me, the good reason that the technology was there developed first. Okay, so we have since the early 2000 where the genome were fully sequenced, then the tools with microarrays starting, and then the sequencing technology was possible to really go into some. First, the cost of the measurements and the amount of data generated was really possible. Now, both at the analytical level, both at the metabolomic level and at the proteomic level, the things are really improving dramatically. I did my PhD using proteomics. At that time, I was doing two-dimensional gel electrophoresis. It was another era, I would say. Now we can think about using proteomics at scale with thousands of molecules, which can be thousands of proteins, which can be monitored very precisely with all the post-translational modification assays as well. And I would say that we need still to be able to compare the biological information one can retrieve for the transcriptomic compared to the proteomics. And adding also the metabolomics also in this evaluation. And honestly, it's part of the objective. One of the objectives of Oasis is to really assess how much we learn from one layer of the biological effects in the transcriptomics, proteomics, metabolomics is not part of Oasis, but and the cell painting. But for me, that is still under discussion. We know there are differences, but we don't have for me a clear picture about the real status of
So now there's many questions.
that.
Now there is a question from David Guignard. So hi David, with cell painting, it seems that the effect is highly dependent on the selected cell line. So it's going to be a big debate. So please be short. So it's highly dependent on the selected cell line, more than with transcriptomics. How do you manage this cell line dependency to extrapolate the in vitro data to human? Please be short, if it's possible. Because
but I can be. It's complicated. It's super complicated. So it's all about first the nature of the effects revealed by using cell painting, and secondly about the nature of the choice of the cell line. Of course, the cell line, and again, to be short and maybe to invite you to go into the outcome of this Oasis project, because in Oasis, we decided to go into the comparison between U2S, which is an osteoblast cell line, to predict liver tox. Okay, so how much an osteoblast cell line can predict liver tox compared to EPIRG, for example. So this type of question we try to answer. Yeah. So this is I don't have definitive answer on that. So there are a lot of things. Part of the limitation is about that.
That's excellent comment. I mean, it was very nicely represented with your data representing the two prostate cancer cell lines. That was, which is only mentioning that prostate cancer is very diverse. Also.
Yeah. Sure.
Then there is another question from Danilo. David, if I recall right, originally Oasis, it's a technical question, was selecting DrugSec as technology for transcriptomic profiling. What was the reason for changing to Tempo-OSEC, other than the fact Tempo-OSEC is widely used in the field?
Alors, actually, we are doing both. So we have different kind of evaluation which are performed with one or the other technology.
Okay. Another question from Davy. Oasis is looking for hepatotoxicity. Are there similar initiatives investigating other type of toxicity?
That's for phase 2 of Oasis, where we are discussing about some stuff which are really again. I said that it was the liver was initially about a compromise, because when I came to, I have initiated this Oasis consortium. So initially, with ACHEM, the first thing I have in mind was to think about doing something related to endocrine aspects, okay, endocrine disruptor topics. But if you discuss with the guys from pharma, endocrine, it's not the primary focus. So they would come and say, yeah, but why not about going into the kidney or the cardiotox and so on and so forth. But for us, kidney is not the. So we landed onto the liver as a compromise, because we do have still some interest in the liver for us in the context of carcinogenicity in particular in the ACHEM sector. And for pharma, it's a big topic also, the liver tox. But the next phase is about expanding both the tox endpoints and probably technology as well.
Okay, now there is a long question from William Bisson. So great talk, David, with important question for reality check. Interesting your slide with chemical and biological space. Is fit for purpose the way to approach focus space of chemistry for a given adverse outcome? And considering the next generation state of the art technology in assays, is biological similarity more relevant than chemical similarity for predictive toxicology and engineering? That's a tough one.
Yeah, but that's the key thing, William. It's I would dare to say, for me, yes, because biology somehow is agnostic of the chemistry. So you look at the effects. You look at what the perturbators. You see, it can even be not a molecule. It cannot be a chemical. It can be any kind of perturbator. So you can predict out of the effects what's going to be the potential of the molecule to induce harm. The tricky thing with that one, compared to the QSAR model, is that it has a cost. The cost is that you need to synthesize the molecule if you are talking about chemistry, and you need to do the experiment in vitro, which is coming with its own constraints about the reproducibility, the technical variability, the biological variability, and the cost. QSAR model, as soon as you get a molecule, on the computer, you can draw the molecule and make use of your new molecule directly in your model. If you do the so-called Q-bar, the quantitative biological activity relationship, instead of the QSAR, the structural one, then you need to go into the hard the hard reality of the experimental data generation. Yeah.
So the last question is from Isabelle Lecomte. Hi, David. What can you say about the application in the developmental toxicology toxicity, which is a very important field?
What I can say, I would say we probably need to wait a little bit because we are working on it, and but there are some work we are performing in the lab with the cell painting, because the key advantage of the cell painting is the throughput and the cost. It's super cost effective. So that's something which, in theory, allows us to really build super large data sets to really populate with many kind of example, as many as possible examples. In theory, with tox, you don't have so much of the constraints in terms of number of chemicals. So a campaign of cell painting, it's something that you can run 100. Yeah, so depending on the number of concentration you're evaluating, it goes from 2 to 300 to almost 800 in our lab. Okay, and we are only running the thing in 384. You can run it in 1536. So we have some evidence that you can make use of the cell painting for in cell lines using U2S to predict developmental tox issues. Yeah.
Okay, thank you very much, David. I think we have finished the time. It's 2 p.m. It's 2:01, so that's perfect. So again, I will tell all the participants that the presentation of David will be available on the YouTube channel of EUROTOX. So thank you again, David, for this very nice presentation. A lot of questions, a lot of interaction. Thank
Okay.
you again. So
Thank you.
everybody
Thank you
have a
to
nice
you.
day. Bye-bye.
Yeah, thank you. Bye-bye.
Bye-bye.